Zombie ant fungus. Ophiocordyceps unilateralis is the infamous fungus that turns ants into real-life zombies. Once spores land on an ant, they break through the exoskeleton and spread through the body while keeping the host alive. The fungus releases chemicals that hijack the ant's nervous system, forcing it to abandon its colony and climb upward. At a precise height, around 25 centimeters above the forest floor, the ant locks its jaws onto a leaf vein in a death grip. This is no accident. The fungus times the death to match the perfect temperature and humidity for spore growth. After the ant dies, a stalk erupts from its head, raining spores onto ants passing below. Scientists discovered the fungus doesn't invade the brain directly, but forms a network around it, releasing chemicals that manipulate behavior with terrifying precision. Entire colonies can be wiped out by this mind-controlling parasite. Cicada Sex Hijacker. Massospora cicadina is a fungus that literally weaponizes the cicada's sex drive. It infects males during their underground larval stage, then waits silently for over a decade. When the cicadas finally emerge, in their 13 or 17-year cycles, the parasite takes over. Instead of killing the host outright, the fungus consumes the cicada's abdomen and replaces it with a chalky mass of spores. Yet the infected male keeps flying, keeps calling, and even mimics female wing-flicking signals to attract healthy males. The fungus goes further, producing amphetamine-like chemicals that keep the cicadas hyperactive and sexually compulsive, even as their bodies crumble. Some infected cicadas lose their entire abdomen, but still attempt to mate, spreading spores with every encounter. It's one of the most disturbing strategies in the insect world, turning reproduction into a delivery system for infection. Mind-controlling liver fluke. Dicrocelium dendriticum hijacks ant brains to get eaten by sheep, completing one of nature's most manipulative life cycles. This tiny fluke needs 3 different hosts to reproduce, and it turns ants into suicidal zombies to reach its final destination inside a grazing mammal's liver. The fluke's journey begins when snails eat contaminated sheep droppings and become infected. The parasites multiply inside the snails, which then cough up slime balls packed with fluke larvae. Ants discover these slime balls and eat them like candy, unknowingly consuming thousands of parasites. Most larvae hide in the ant's body, but one special larva travels directly to the brain. This brain fluke doesn't kill the ant, it enslaves it. When evening temperatures drop, the brain parasite forces the ant to climb grass blades and clamp its jaws onto the tips, making it an easy target for grazing sheep. If the ant survives the night, it returns to normal behavior until the next evening's mind control session. Once eaten, the flukes travel to the sheep's liver where they reproduce, starting the cycle again with contaminated droppings. Pulsating snatchers. Leucochloridium paradoxum transforms snail eye stalks into throbbing, colorful caterpillar mimics that birds can't resist eating. These parasitic flatworms have evolved one of the most visually disturbing takeover strategies in nature, turning their host into a living lure. The parasite begins its life cycle when birds infected with adult worms drop contaminated feces near snails. When snails eat the droppings, the parasites hatch and migrate throughout the host's body. The most sinister larvae make their way to the snail's eye stalks, where they begin their grotesque transformation. The infected eye stalks swell dramatically and develop bright green and orange bands that pulse rhythmically like breathing caterpillars. The parasite even controls the snail's behavior, forcing it to climb into bright sunlight where the fake caterpillars are most visible to hungry birds. The pulsing motion is so realistic that birds immediately attack the eye stalks, swallowing the parasites. The snail often survives losing its eyes, but the parasites complete their journey inside the bird's digestive system, ready to start the cycle again. Hairworm water suicide. Spinochordotes tillinii drives crickets and grasshoppers to drown themselves in water, emerging from their bodies like horrifying alien creatures. These parasitic worms can grow longer than their hosts and need water to reproduce, so they literally brainwash insects into committing aquatic suicide. The hairworm begins as a microscopic larva that crickets accidentally consume while drinking from puddles or eating infected insects. Once inside, the parasite grows into a writhing worm that can reach 4 times the length of its host's body, coiled up inside the cricket's abdomen. The worm produces proteins that alter the cricket's brain chemistry, specifically targeting neurons that control thirst and movement. When ready to reproduce, the parasite hijacks the cricket's navigation system and forces it to seek water, any water. Infected crickets will leap into swimming pools, streams, or even water bowls, drowning themselves in the process. The moment the cricket hits the water, the hairworm bursts out like a scene from a horror movie, writhing freely as it searches for a mate. Studies show infected crickets are 20 times more likely to end up in water than healthy ones, proving the parasite's mind control is devastatingly effective. Fish behavior hijacker. Schistocephalus solidus turns tiny sticklebacks into reckless daredevils that practically beg to be eaten by birds. This tapeworm grows so large inside fish that it can make up 40% of the host's body weight, while simultaneously rewiring the fish's survival instincts to ensure its own reproduction. The parasite parasite starts as eggs in bird droppings that contaminate water where they're eaten by tiny crustaceans called copepods. When sticklebacks eat infected copepods, the tapeworm larvae burrow into the fish's body cavity and begin growing at an alarming rate. The massive parasite steals nutrients so efficiently that infected fish often become visibly bloated and struggle to swim normally. But the physical burden is nothing compared to the behavioral manipulation. The tapeworm produces hormones that make infected fish lose their natural fear responses and become hyperactive near the water's surface, where birds hunt. Healthy sticklebacks hide in vegetation and flee from shadows, but infected fish swim boldly in open water, practically advertising themselves to predators. When birds finally eat these reckless fish, the tapeworms mature in their new host's intestines, completing their journey from water to bird and back again. Killifish surfing parasite. Euhaplorchus californiensis transforms killifish into surface-skimming daredevils that flash like disco balls to attract bird predators. This microscopic fluke invades the fish's brain and literally makes them shimmer and dance at the water's surface, turning their natural camouflage into a death sentence. The parasite begins when birds infected with adult flukes defecate into California salt marshes. Snails eat the contaminated waste, and the parasites multiply inside them before being released as swimming larvae called cercariae. When these microscopic invaders encounter killifish, they burrow directly into the brain tissue and form protective cysts around themselves. Once established in the brain, the parasites cause infected fish to lose their schooling behavior and swim erratically near the surface. The most dramatic change is the flashing behavior. Infected killifish repeatedly roll sideways, exposing their silver belly scales that reflect sunlight like mirrors. This creates an irresistible target for diving birds who can spot the flashing fish from hundreds of feet away. The parasite essentially turns the fish into a living fishing lure, guaranteeing its transmission to the final host, where it reproduces in the bird's intestines. Eye-clouding vision thief. Diplostomum pseudospathicium blinds fish by growing inside their eye lenses, then forces them to swim in shallow water where birds can easily snatch them. These parasites literally fog up their host's vision while manipulating their behavior to maximize the chances of being eaten by the right predator. The fluke's life cycle begins in waterbird intestines, where adult parasites release eggs that contaminate freshwater environments. Snails become the first intermediate host, harboring developing parasites that eventually emerge as free-swimming larvae. When these larvae encounter fish like trout or perch, they penetrate the skin and migrate directly to the eyes. Inside the eye lens, the parasites form visible white spots that increasingly cloud the fish's vision as they grow. Partially blinded fish can't see predators approaching from above, but the parasite goes further by altering their behavior. Infected fish spend more time in shallow water and near the surface, exactly where diving birds hunt most effectively. The combination of impaired vision and risky behavior makes infected fish sitting ducks for predators, allowing the parasites to complete their journey back to bird intestines, where the cycle begins anew. Cockroach brain surgeon wasp. Ampulex compressa performs precise brain surgery on cockroaches, injecting venom that turns them into docile zombies for its larvae to eat alive. This jewel wasp has evolved surgical precision that would make neurosurgeons jealous, delivering targeted toxins to specific brain regions with just 2 stings. The emerald jewel wasp hunts cockroaches 3 times its size using a technique perfected over millions of years. The first sting paralyzes the cockroach's front legs temporarily, allowing the wasp to position itself for the critical second strike. The wasp then inserts its stinger directly into the cockroach's brain and injects a cocktail of neurotoxins that target the escape reflex center. The zombified cockroach remains conscious and can walk normally, but loses all motivation to flee or defend itself. The wasp literally leads its victim by the antenna like a dog on a leash to a prepared burrow. After laying a single egg on the cockroach's leg, the wasp seals them both underground, where the hatching larva feeds on the living but helpless cockroach for weeks. The venom is so precisely calibrated that the cockroach stays fresh and alive throughout the entire feeding process, providing the ultimate meal for the developing wasp. Spiderweb architect wasp. Hymenopemisis argyrophaga hijacks spider brains to force them to build custom death traps for the wasp's offspring. This parasitic wasp doesn't just control spider behavior, it reprograms their web-building instincts to construct completely unnatural structures designed for larval protection. The adult female wasp hunts orb weaver spiders and temporarily paralyzes them with a precise sting. While the spider is immobilized, the wasp glues a single egg to its abdomen, then flies away, leaving the spider apparently unharmed. The spider continues its normal life for 2 weeks, building regular webs and catching prey, completely unaware of the larva growing on its back. When the larva is ready to pupate, it injects chemicals into the spider that completely override its web-building program. Instead of constructing a normal spiral orb web, the mind-controlled spider builds a bizarre cocoon web, a small, incredibly strong structure unlike anything it would naturally create. After completing this custom nursery, the spider dies, and the larva spins its cocoon inside the protective web. The manipulated architecture is so effective that the cocoon survives storms and predators that would destroy normal spider webs. Caterpillar Bodyguard Hijacker. Glyptopontylus wasps turn caterpillars into suicidal bodyguards that violently defend the wasp larvae feeding on their siblings. These parasites pull off one of nature's most twisted schemes, convincing their host to become a loyal protector, even while being eaten alive from the inside. Up to 80 wasp larvae develop inside a single tobacco hornworm caterpillar, growing fat on the host's body fluids while somehow keeping it alive and healthy. After 2 weeks of internal feeding, the mature larvae chew exit holes through the caterpillar's skin and emerge like a scene from Alien. They immediately begin spinning cocoons on nearby leaves, apparently safe from the host they just abandoned. But the caterpillar doesn't die or wander away. It becomes a ferocious guardian. The remaining wasp larvae in its body pump out chemicals that turn the caterpillar into an aggressive sentry that violently headbutts any predator approaching the cocoons. The zombified caterpillar stops eating and spends its remaining days thrashing at beetles, ants, and other threats trying to attack the developing wasps. When the adult wasps finally emerge from their cocoons, their former host dies, having sacrificed its life protecting the very parasites that consumed it. Hornworm Mind Controller. Cotesia congregata transforms tobacco hornworm caterpillars into living nurseries, then reprograms them to become violent protectors of their own parasites. These wasps have mastered the art of keeping their hosts alive while simultaneously preparing them for a gruesome role reversal. Female wasps inject dozens of eggs directly into young hornworm caterpillars, along with a cocktail of viruses and chemicals that suppress the host's immune system. The wasp larvae grow inside the caterpillar's body cavity, feeding on its blood and tissues while carefully avoiding vital organs. The infected caterpillar continues eating and growing normally, unknowingly nourishing an army of parasites. When ready to pupate, up to 100 wasp larvae simultaneously burst through the caterpillar's skin and begin spinning white cocoons on nearby vegetation. The host should die from this trauma, but instead remains alive and undergoes a dramatic personality change. The dying caterpillar becomes hyper-aggressive, rearing up and thrashing violently at any creature that approaches the cocoons. This bodyguard behavior is so effective that wasp survival rates increase dramatically when their former host remains alive to protect them during their vulnerable pupal stage. Ladybug Zombie Guardian. Dinocampus coccinelle turns ladybugs into paralyzed bodyguards that stand motionless over wasp cocoons like living umbrellas. This wasp larva develops inside the ladybug for 3 weeks, then emerges and spins its cocoon directly beneath the host, which becomes a protective shield until the adult wasp hatches. The female wasp injects a single egg into an adult ladybug, along with a virus that prevents the host immune system from attacking the developing larva. The infected ladybug behaves completely normally for weeks, while the wasp larva feeds on its body fluids from the inside. The host continues hunting aphids and mating, showing no signs that a parasite is consuming it alive. When the larva is ready to pupate, it chews an exit hole through the ladybug's leg joints and emerges to spin a yellow cocoon directly underneath its host. Instead of dying or flying away, the ladybug becomes frozen in place, standing guard over the cocoon with its legs spread wide like a protective tent. The paralyzed ladybug can't move or feed, but it trembles violently when predators approach, scaring away ants and other threats. About 25% of ladybugs survive this ordeal and eventually recover after the adult wasp emerges, making this one of the few parasites that sometimes lets its host live. Cat Parasite Behavior Hacker. Toxoplasma gondii rewires mammal brains to eliminate their fear of cats, turning natural prey into willing victims for the parasite's reproductive strategy. This single-celled organism has infected an estimated 1/3 of all humans on Earth and subtly alters personality and decision-making in ways scientists are still discovering. The parasite can only reproduce sexually inside cat intestines, but it infects virtually every warm-blooded animal as intermediate hosts. When rodents become infected by eating contaminated cat feces, the parasite travels to their brains and performs a stunning act of behavioral manipulation. Infected mice and rats lose their innate fear of cat odor and actually become attracted to the smell of cat urine. This fatal attraction ensures infected rodents get eaten by cats, allowing toxoplasma to complete its life cycle. But humans aren't immune to the parasite's influence. Studies suggest infected people become more impulsive, have slower reaction times, and show altered personality traits. Some research even links toxoplasma infection to increased risk-taking behavior and higher rates of traffic accidents. The parasite essentially turns mammals into cat-seeking missiles, manipulating ancient fear circuits that evolved to keep prey animals alive. Bee colony death driver. Nosema apis and Nosema serrana destroy bee colonies from within by hijacking worker bee intestines and turning them into spore-producing factories. These microsporidian parasites cause explosive diarrhea in bees while manipulating their behavior to spread the infection throughout entire hives, contributing to devastating colony collapse events. When bees consume contaminated water or food, Nosema spores germinate in their midgut and begin multiplying rapidly. The parasites damage intestinal walls so severely that infected bees develop chronic diarrhea, spreading millions of spores throughout the hive. The infection disrupts nutrient absorption, leaving bees malnourished and weakened despite normal feeding behavior. Infected worker bees undergo disturbing behavioral changes that accelerate colony destruction. They become hyperactive and disoriented, often abandoning the hive prematurely and dying far from home. The parasites also manipulate infected bees to defecate inside the hive instead of flying outside, contaminating food stores and spreading infection to healthy nestmates. Queen bees infected with nosema lay fewer eggs and die earlier, while infected workers produce less royal jelly to feed developing larvae. Entire colonies can collapse within months as the parasite spreads exponentially through the social network. Crab gender bender. Sacculina carcini chemically castrates male crabs and forces them to behave like protective mothers, caring for the parasite's own offspring. This parasitic barnacle performs one of nature's most complete takeovers, essentially turning its host into a living nursery while stealing its entire reproductive future. The parasite begins as a free-swimming larva that seeks out shore crabs and injects itself through microscopic gaps in their shell joints. Once inside, Sacculina grows throughout the crab's body like a root system, absorbing nutrients while carefully avoiding vital organs. The parasite eventually emerges as a yellow sac beneath the crab's abdomen, exactly where the crab would normally carry its own eggs. Male crabs undergo the most dramatic transformation. The parasite pumps them full of feminizing hormones that cause their claws to shrink, their abdomens to broaden, and their behavior to change completely. Infected males stop fighting other crabs and begin exhibiting maternal behaviors, carefully tending and protecting the parasite's egg sac as if it were their own brood. They even fan the parasite's eggs with their swimming appendages and defend them aggressively from predators. The chemical castration is so complete that infected crabs never reproduce again, dedicating their entire lives to raising the parasite's offspring. Rage virus brain hijacker. Rabies virus transforms peaceful mammals into aggressive, biting machines by systematically destroying brain regions that control fear and rational behavior. This neurotropic virus has evolved the perfect strategy for transmission, turning its dying hosts into violent spreaders that bite anything within reach during their final days of life. The virus enters through bite wounds and travels along nerve pathways directly to the brain, avoiding the immune system's detection for weeks or months. Once it reaches the central nervous system, rabies systematically attacks the limbic system, particularly the hippocampus and amygdala that regulate emotion and aggression. Infected animals lose their natural fear responses and become hyper-aggressive, attacking humans, pets, and even inanimate objects. The virus's most sinister manipulation involves hydrophobia, an intense fear of water that develops in later stages. This isn't just a symptom, it's a calculated strategy. Infected animals avoid drinking water, concentrating the virus in their saliva to maximum infectious levels. The combination of aggressive biting behavior and hyper-concentrated viral loads in saliva makes rabies one of the most efficient transmission systems in nature. Once symptoms appear, the infection is virtually 100% fatal, making rabies the deadliest parasite on this list. Bird navigation destroyer. Borna disease virus infiltrates bird brains and scrambles their internal GPS systems, causing infected birds to crash into buildings, fly in circles, or become completely lost during migration. This RNA virus specifically targets neurons responsible for spatial orientation and learning, turning master navigators into helpless wanderers. The virus primarily infects songbirds, waterfowl, and raptors by spreading through contaminated food, water, or direct contact with infected birds. Once inside the brain, Borna virus attacks the hippocampus and other regions crucial for memory formation and spatial navigation. Infected birds lose their ability to form mental maps of their territory and can't remember familiar landmarks or roosting sites. The behavioral changes are heartbreaking to observe. Infected birds repeatedly fly into windows, trees, and buildings as their collision avoidance systems fail. During migration season, infected flocks become separated as individual birds veer off course or stop flying altogether. Some birds develop repetitive behaviors like endless circling flights or obsessive head movements. The virus doesn't usually kill directly, but the navigation impairment leads to starvation, exhaustion, and fatal accidents. Entire bird populations can be devastated when Borna virus spreads through roosting sites or migration stopover points. Insect reproduction hijacker. Wolbachia bacteria infects up to 70% of all insect species and manipulates their reproduction through 4 different strategies: killing males, turning males into females, forcing virgin births, and making infected females incompatible with uninfected males. This bacterial parasite has become one of evolution's most successful manipulators by controlling how insects reproduce. The bacteria lives inside insect cells and spreads only through infected mothers to their offspring, creating enormous pressure to bias reproduction toward females. In some species, Wolbachia simply kills all male embryos, forcing infected mothers to produce only daughters. In others, it feminizes male insects by interfering with sex sex-determining hormones, creating functional females that carry and spread the infection. Perhaps most deviously, Wolbachia performs cytoplasmic incompatibility. Infected males can only successfully reproduce with infected females, while matings between infected males and uninfected females produce dead offspring. This forces entire populations to become infected or face reproductive failure. In some butterfly species, Wolbachia allows virgin females to reproduce without males at all, creating entire populations of clones. Scientists estimate this single bacterial genus may have shaped insect evolution more than any other factor, making it the ultimate reproductive puppet master. Tick disease vector. Rickettsia bacteria transforms ticks into disease-spreading weapons by manipulating their feeding behavior and making them more aggressive toward larger hosts. These intracellular parasites cause Rocky Mountain spotted fever and other deadly diseases while rewiring tick behavior to maximize transmission opportunities. Rickettsia infects tick cells throughout their body, including salivary glands, ovaries, and gut tissues. The bacteria can pass from infected female ticks to their eggs, ensuring the next generation is born already infected. When infected ticks feed on blood, the bacteria multiply rapidly in their salivary glands and get injected into the host along with anticoagulant saliva. Infected ticks exhibit more aggressive host-seeking behavior, climbing higher on vegetation and remaining active for longer periods to encounter passing mammals. The bacteria also prolongs tick feeding time by interfering with the host's immune responses, allowing more time for bacterial transmission. Once inside mammalian hosts, Rickettsia invades blood vessel walls and causes internal bleeding, organ failure, and death if untreated. The bacteria essentially turns ticks into biological warfare agents, using them as both vehicles and weapons to spread deadly infections across entire ecosystems.